Hello, I am Professor Sims, and in this video we will be discussing Proteobacteria, gram-negative bacteria, gram-positive bacteria, Phototrophics, and Archaea. The learning objectives for this unit include identifying and describing unique examples of prokaryotes, explaining how prokaryotes are classified and what kind of features they have, describing unique features of non-proteobacteria, gram-negative bacteria, and identifying, describing unique features of phototrophic bacteria. We're going to compare and contrast high GC and low GC bacteria. We're going to give examples of deeply branching bacteria and features of Archaea, giving examples of unique habitats of Archaea. As y'all know by now, prokaryotes are unicellular. They have no nucleus. They're ubiquitous. They're found almost everywhere on the planet, including some very extreme environments. We've talked about different extremophiles and halotolerant bacteria. Prokaryotes can adjust their feeding habits to available natural resources, which is an evolutionary benefit that they enjoy. They live in communities and they can interact with hosts. Prokaryotes living in or on the human body. That is what is known as the human microbiome. And this varies between individuals. It also varies between different parts of the body, and it can change over time. But it is very significant because the human microbiome, honest to goodness, you have 10 times as many bacterial cells in your body than you do human cells, which is staggering. The total prokaryotes within a region of the body, for example, your mouth or in your throat or in your stomach, that's the microbiota of that region. Different environments where prokaryotes can live. This here is the Dead Sea, and yes, there's plenty of halotolerant and halophile bacteria that do very, very well in the Dead Sea. This Archaea, Halobacterium solanarum, is a halophile that lives in the Dead Sea. Shoamella lives in the deep sea, and it can live in very, very low oxygen. It survives very harsh environment by attaching to the seafloor, and it has these things called nanocables that it uses to sense oxygen, so it can kind of seek out any oxygen that is diffused in the water way down on the bottom of the ocean. Prokaryotes are classified into domains Archaea and bacteria. Microorganisms are found in all three kingdoms, but prokaryotes inhabit the kingdom of bacteria and Archaea. And of course, eukarya is where eukaryotes live, and of course eukarya is also where plants and animals, including ourselves, live in the kingdom eukarya as well. Proteobacteria is a phylum first discovered by Carl Wise based on nucleotide sequences. They're classified as alpha, beta, gamma, delta, or epsilon, and they all have different orders, family, genre, and species within those classifications. Alpha proteobacteria are what is known as oligotrophs. They can convert atmospheric nitrogen into nitrites, which is very useful for farming because most other life forms cannot convert atmospheric nitrogen and use them for nutritional purposes, but alpha proteobacteria can. Beta proteobacteria are eutrophs, and they do include some human pathogens of the genus Neisseria and the species Bordetella pertussis. And I just noticed I got a typo here because genus and species names should be in italics. So yeah, it trips me up sometimes too, you guys. Gamma proteobacteria are the most diverse group of proteobacteria, include aerobic and or facultative anaerobic human pathogens. So here's an example of some nitrogen-fixing bacteria. These are rhizobium that live in root nodules of legumes, and they give an example of clover. And this is electron microscope image, a micrograph of a root nodule that shows the bacteroids, bacterium-like cells or modified bacterial cells that live within the root nodule. It's kind of a symbiotic relationship that they have. The rhizobium is converting the nitrogen from the atmosphere into nitrite, and then the legume plant can use the nitrite to make energy. This is Neisseria meningitidis, agent that causes meningitis. Here we'll go through some notable examples of proteobacteria. First we have chlamydia. This is an obligatory intercellular bacteria. Some but not not all, cause chlamydia, trachoma, and pneumonia. Rhizobium, this is a nitrogen-fixing bacteria, so he's actually a beneficial species or genus. Rhizobium, this is a genus that is nitrogen-fixing bacteria that live in the soil, so these are actually beneficial proteobacteria. Next we have Rickettsia. This is an alpha proteobacteria that is associated with the Rocky Mountain spotted fever and typhus. He is transmitted by ticks. Bordetella, you guys have heard of this guy. He is a beta proteobacteria. He's very fastidious. He is responsible for causing pertussis, whooping cough. Let's see, Enterobacter. Enterobacter causes urinary and respiratory tract infections. Escherichia is a facultative anaerobe. He's a gamma proteobacteria. He is often used as a model organism for genetics and molecular biology. Skip around a little bit here. We've got Klebsiella. He's a gamma proteobacteria as well. He causes pneumonia, and he's a facultative anaerobe. Legionella is the causative agent of Legionnaire's disease. Proteus is associated with urinary tract infections and sepsis. This is especially true for hospital-acquired infections. He's also a gamma proteobacteria. Pseudomonas is a gamma proteobacteria that is antibiotic resistant and is associated with many hospital-acquired infections. Shigella is a gamma proteobacteria. It produces the Shiga toxin, which causes dysentery. Vibrio is gamma proteobacteria. One of the species of Vibrio is Vibrio cholera that causes cholera. And Yersinia is the causative agent of bubonic plague and the pneumonic plague, and can be associated with severe diarrhea in humans. And that is another gamma proteobacteria. Next, we have some delta and epsilon proteobacteria. These are represented here by Desulfovibrio, which is a delta proteobacteria. He can actually be used as a beneficial bacteria. He removes toxic and radioactive waste, so that is a very beneficial microorganism. Then you have Myxobacterium, which is another delta proteobacteria. He usually lives in soil, and he's used as a model organism for studies having to do with intercellular communication, like quorum signaling and biofilm production. Campylobacter, he's known for causing severe enteritis. And then Helicobacter causes chronic gastritis, peptic ulcers, and stomach cancer. He is an epsilon proteobacteria. Some gram-negative non-proteobacteria include spirochetes, cytophaga, Fusiobacterium, Bacteroides, Planktonomyces, and many phototrophic bacteria. Spirochetes are motile, spiral-shaped bacteria with a long, narrow body. Several spirochete genera include human pathogens, and some of these are your syphilis and Lyme disease. One gamma proteobacteria is Escherichia coli, E. coli. E. coli is perhaps the most studied bacterium out of all of them. Delta proteobacteria can reduce sulfate and elemental sulfur. Delta proteobacteria, some of them are scavengers, and they form myxospores and fruiting bodies. So I'll show you a figure of that in just a moment. Epsilon proteobacteria are the smallest group of proteobacteria. They do include Campylobacter and Helicobacter, which are human pathogens. This is Myxobacteria, fruiting bodies. And this is Helicobacter pylori, which causes chronic gastritis and can lead to ulcers and even stomach cancer. So he's a bad guy. Some examples of non-proteobacteria and phototrophic bacteria. Here we have Bacteroides. He is a mutualistic non-proteobacteria. He is associated with several opportunistic pathogens. Cytophaga is a non-proteobacteria that decomposes cellulose and is known to cause disease in several fish species. Fusobacterium is non-proteobacteria that is anaerobic and forms biofilms. It can cause certain diseases in humans, including periodontitis, which is gum disease, plaques, and it can also cause ulcers in humans. Treponema is a non-proteobacteria that causes syphilis. Cyanobacteria is a phototrophic bacteria. He's oxygenic and is one of the blue-green bacteria. An example is M. aeruginosa. Then you have Chlorobe. He is a phototrophic anoxygenic green sulfur bacteria. Then you have Chloroflexi, which is anoxygenic and another green sulfur bacteria. And then purple non-sulfur bacteria. So you have some species that are in the same genus that can be in different groups. It's kind of confusing, but I'm going to tell you these are going to be limited to your Rhodosporidium, Rhodocyclus, and Chromatium. So these are your phototrophic proteobacteria. Spirochetes, Cytophaga, Fusobacterium, Bacteroids, Plantomycetes, and many phototrophic bacteria. Not all, but many. Spirochetes are modal, which means they can move, and they're spiral shaped. Those are the ones that have like the true corkscrew shape. They have long, narrow body. Some spirochete genera include human pathogens, for example, syphilis and Lyme disease. This is some dark field microscopy and some electron microscopy looking at spirochetes. You can see here that they have like a they're not just spirilia, they have a corkscrew cell morphology. This is a cross section here where it's showing the axial filaments and then the cell membranes. And the axial filament is this bit that goes around. It's kind of the part that gives it a corkscrew twist. Cytophaga, Fusobacterium, and Bacteroides, they're grouped into what is known as the CFB group. They're all rod-shaped, they're all anaerobic, and they're all organoheterotrophs, and they are avid fermenters. Cytophaga are aquatic, and they are very modal. Fusobacteria, they live in your mouth, and they can cause severe infectious diseases. Bacteroides, these guys generally are non-pathogenic. Most of them are mutualistic. A lot of them are normal flora of the gut, but they can become pathogenic just like any other normal flora can become pathogenic if they are introduced to places where they shouldn't be, right? Like the bloodstream. Planktonomyces are aquatic and they reproduce by budding, and they can form very large colonies. Figure 414, this is Bacteroides, up to 30% of the normal microbiota of the human gut. So these guys are very well acquainted with us, and usually they don't do us any harm. This is an example of sessile planktonomyces, and again, sessile just means that it is permanently attached. So they have what is named a hold fast that allows them to, you guessed it, hold fast in aquatic environments. And that can be important because the water is always trying to slosh you around and move you around. But they stand their ground with these attachments that they make. But they do have swarmers of planktonomyces that, because this is a genus and there are different species of it. And some of them are swarmers that move around quite a bit. They're quite modal. Phototrophic bacteria use the energy from the sun, and they are separated into groups called sulfur bacteria and non-sulfur bacteria. Sulfur bacteria are anoxygenic photosynthesizers. They use sulfur as electron donors. Non-sulfur bacteria use organic compounds as electron donors for photosynthesis. Some phototrophic bacteria can fix nitrogen, again, which is very beneficial for them. And cyanobacteria are oxygen-producing bacteria. And these guys are important because they're thought to be very critical in the formation of Earth's atmosphere. So here, figure 416, this is showing an example of some of your sulfur bacteria. They generally have this either purplish color or greenish color, and they do use bacterial chlorophylls to perform photosynthesis using sulfur as an electron donor. Examples of cyanobacteria generally are found in freshwater and in warm temperatures over here. Microcystis aeruginosa and some other cyanobacteria that have multiplied rapidly, and they have the added pleasantry of producing neurotoxin, which of course is harmful to fish and other aquatic animals. Gram-positive is a very large, diverse group, and this table gives some examples of your gram-positive high GC and low GC bacteria. So you have your actinomyces. This one is associated with gum disease. Your Corynebacterium causes diphtheria, micrococcus. There are some that are opportunistic pathogens, but these are rather ubiquitous in the environment, and a lot of them are non-pathogenic. Then you have Mycobacterium. Your Mycobacterium are very famously known for causing tuberculosis and leprosy. Streptomyces, some species of Streptomyces are spore-forming. They are decomposers in nature, so they can be beneficial. They're also beneficial in the fact that they're used for synthesizing antibiotics. You have Bacillus. Most species of Bacillus form endospores. One species of Bacillus is actually the causative agent of anthrax. And Bacillus cereus is generally non-pathogenic, but it can cause food poisoning. Clostridium, he's a low GC gram-positive bacteria. All known species of Clostridium are pathogenic to humans. Some of them cause tetanus, gas gangrene, botulism, and colitis. Enterococcus is again a low GC gram-positive bacilli, and he is associated with urinary tract and other nosocomial infections. Lactobacilli is a low GC bacilli, gram-positive bacilli. Lactobacillus are facultative anaerobes, and they ferment sugars. They are normal flora of the vaginal microbiota, and they are used as probiotics. They're also used in the fermentation of certain food items. They're used to make yogurt, for example. Mycoplasma is a low GC gram-positive bacilli, and these are kind of unique because they have no cell wall, but they're classified as low GC gram-positive because of their genome. And one species of Mycoplasma pneumoniae, is the causative agent of walking pneumonia. Staphylococcus. This has Staphylococcus listed as bacilli. I think that is a typo, as well as Streptococcus below. These are not bacilli, they are cocci. However, they are low GC gram-positives. Staphylococci are unique in that they are halotolerant, and they produce catalase. They also produce coagulase, and they have some toxins that are responsible for local skin and generalized infections. Not all Staphylococci are pathogenic, but the ones that you know of are the ones that cause staph infections. Streptococcus is a really diverse genus, and they produce certain antigens. Some cause hemolysis. Some cause toxins that are responsible for human infections of the throat and other generalized diseases. So one notable species of Streptococcus that is human pathogen is your Streptococcus pyogenes. That's what causes strep throat. Gram-positive, because they have the very thick layer of peptidoglycan, are grouped into high GC and low GC gram-positive. The GC stands for guanine and cytosine, which are nucleotides, right? So DNA has guanine, cytosine, adenine, or thymine nucleotides. Actinobacteria are the name given for high GC gram-positive bacteria, and this includes Actinomyces, Arthrobacter, Corynebacterium, Frankia, Gardnerella, Micrococcus, Mycobacterium, Nocardia, Proteobacteria, Rhodococcus, and Streptomyces. Some of your actinobacteria are used in industry, while others are human and/or animal pathogens. Some pathogenic actinobacteria are really nasty, some of the worst plagues in human history. You've got your Mycobacterium tuberculosis, which causes tuberculosis, and then the one that causes leprosy is Mycobacterium leprae, and then diphtheria is caused by Corynebacterium diphtheriae. Figure 417 shows some examples of actinomyces and Corynebacterium. So these are all gram-positive, high GC gram-positive bacteria, and this guy right here in the middle is your Corynebacterium diphtheriae, which causes diphtheria. This guy over here, he's a little bit tricky because he is what is known as gram variable, which means it's difficult to get an accurate gram stain, which makes it difficult to diagnose. But this is from a pap smear, and it's showing bacterial vaginosis. Clostridia are examples of low GC gram-positive bacteria. They are obligate anaerobes, which means that they don't survive in oxygen, but they can form endospores in order to survive in oxygen. Oh, I guess I just totally messed that up. But yeah, they can't grow and reproduce in oxygen, but they can survive the harsh condition by forming endospores and essentially hibernating. Yeah, we've talked about that before. Pathogens in the genus Clostridia include the ones that cause gas gangrene, tetanus, and botulism. So Clostridium tetani, Clostridium botulinum, these are nasty, nasty, nasty bacteria. Lactobacillus, these are your Enterococcus, Lactococcus, Leuconostoc, and Streptococcus. Streptococcus, this is the thing, this is what gives you strep throat. Other diseases caused by Streptococcus can be scarlet fever, rheumatic fever, and other respiratory infections. Bacilli are low GC gram-positive bacteria, and they include rod-shaped and cocci. Most bacilli are named for their shape, so they're bacilli-shaped, but wouldn't you know it, there are some cocci-shaped species in the bacilli group. And then some other low GC gram-positive species are your Staphylococcus. Bacillus anthracus is the guy that causes anthrax. And then Bacillus cereus, he's generally pretty harmless, but he can become an opportunistic pathogen in the gastrointestinal tract. We do not work with Bacillus anthracus in the lab. We do work with Bacillus cereus in the lab. He's much safer to work with. Staph aureus, he causes a wide range of different infections. Staph aureus, in fact, is the #1 cause of nosocomial infections, lives in the upper respiratory tract, normal flora. There are different strains of Staph aureus, and some of them are highly pathogenic, and some of them are highly resistant to antibiotics as well. Mycoplasma, these are low GC gram-positive bacteria that lack cell walls. An example of mycoplasma is Mycoplasma pneumoniae, which causes an atypical pneumonia, very problematic pneumonia. Deeply branching bacteria are phylogenetically the most ancient forms of life. Not the most ancient bacteria, forms of life on Earth. They're the closest thing that we've ever been able to find to the universal common ancestor in the phylogenetic tree. So they're kind of a big deal in that regard. They include species that thrive in extreme environments, which makes sense because as ancient as they are, most of them came to be and evolve in conditions that were very extreme when the Earth was first forming its atmosphere and the like. Figure 425, this is Deinococcus radiodurans. They call him Conan the bacterium, and he survived some of the harshest conditions on Earth, including extreme heat, extreme salt, even radiation. Really, really tough guy. Conan the bacterium. Archaea, they're unicellular, they're prokaryotic, and they do differ from bacteria in many aspects. A lot of them are extremophiles. They like extreme temperatures and extreme salinity, salt. Only Archaea are known to produce methane. Nothing else produces methane from nothing. Now, lots of things can convert other gases to methane. Archaea produce methane. Halophilic archaea, they like salt that is close to saturation. So this is a really high salt level. And they perform photosynthesis using bacteriorhodopsin. Based on fossil evidence, again, Archaea, we're talking about some of the oldest organisms on Earth. Archaea do not live in great numbers as far as the human microbiome goes, and they are not known to cause disease. So these very, very, very old, ancient organisms, they don't cause human disease. And you know what? That is really connected to the fact that they like to live in extreme environments. The human body is not an extreme environment. Most of our body is made up of neutral pH, middle range temperature, very, very good for a mesophile or a halotolerant species, but not a halophile, not something that likes high temperatures or high salt or low pH. So that makes sense. Sulfolobus is an archaean. He oxidizes sulfur and can store sulfuric acid. Sulfuric acid will burn your face off, but this guy, not a problem. And then these are halobacteria that are growing in salt ponds. The people that are farming this salt didn't exactly want this archaea living here, but you know, some of them got in there and they're doing just great. They love the salt. They love all of that salt. That is going to do it for the lesson 4 material, and I want to thank you guys for watching. Don't forget to do the reading. Check the description below for more videos related to the topics we covered in this lesson, and leave your comments and questions for me in the comments section below.